Hi guys, so hopefully you have had an opportunity to look through the PowerPoint and you've managed to have a go at the starter task here. So let's have a look at what we expect the answer to be. So if we're looking at draw the generic structure of the amino acid, sorry, I'll just get my little board thing straight. So you should have your carbon attached to the carbon, attached to the nitrogen. We've got the double bond, and then we've got the OH here. The middle central carbon has the hydrogen and then the side chain attached. Remember, the side chain can be any of those branches of the 20 amino acids we know, and two H's here. And the second part of the task was to label the amino and carboxyl group. So remember, the NH2 here is our amine group, so here's our amino group, and the carboxyl group is at the other end of the molecule, isn't it? So the carboxyl group is there. Okay, so if you made an error there, then obviously go back and correct. You do need to know that for your exam, so do make sure that you put in the effort to memorise it. So what we're going to look at in this session is the protein structure and bonding. So I'm going to talk through some of the ideas. It's up to you whether you choose to make notes as we go along, or whether you want to just watch and listen and then go back to the PowerPoint in Teams so that you can then make notes straight from the PowerPoint once you've kind of watched it through once. I'll let you decide how that best works for you. All right. So we're going to build on what we did last time. So we talked about peptides, peptide bonds, and how they join together, and that we can get different sorts of structures. So you may remember that there are so many different types of protein, but all of them are made from just 20 naturally occurring amino acids, and the amino acids can join together into a long chain. So if we've got 100 amino acids in the chain, the combination of amino acids is 20 to the power of 100. So all those different combinations that you can get is going to lead to a huge variety of type of protein that we can get. So the level of protein structure. So you in your exam need to be able to describe the levels, the tiers of structure and what they are in a protein. So the primary structure, so the first level, if you like, is just the sequence. Okay, so it's just the amino acid chain. So if I bring this picture over here so that you can just have a look at it, so you can see that here we've got a series of amino acids, so the green, orange, blue, red, yellow, green, red, blue, but you get the idea. So this is the primary structure, it's just the sequence. The sequence here is green, red, blue, red, yellow. Okay, so that's the first bit. Let's just move you out of the way, little picture. The secondary structure, so the second tier of structuring, is how it folds. So as it's created, as it comes out of the ribosome, and you've got this long chain of amino acids, the secondary structure is what happens to it immediately. How does that begin to fold itself around? Then we've got the next level. So once it's started to fold around, we get different shapes, and then those different shapes need to fold around themselves, and how they then fold around to give the 3D structure of the whole protein, that's called the tertiary structure. And we'll talk a little bit about the bonding that holds both the secondary structure in place and then the tertiary structure in place. The final level, so we don't have necessarily the tertiary and quaternary structure in every single protein. It will depend on the protein that we're making, which we'll talk about in the next session. The final tier of structuring is the quaternary structure, and that's when you get different chains interacting with each other. So hemoglobin is an example of that. Hemoglobin is made of 4 polypeptide chains that interact with one another to form this very large protein. So if we just go back and have another look at the picture here, so you can see what I mean. So here's our secondary structure. In this particular one, what's happened is the chain has twisted on itself to form an alpha helix. Here's the alpha helix in here, and we can see that the alpha helices are then twisted around each other to form this unique structure. This is the tertiary structure. And then these guys can then join up with other ones to form the quaternary structure, and they're sometimes held together or they often interact then with other elements inside as well, which we'll come to. Okay. So that's our tiering. So we're going to talk about it in just a little bit more detail. All right. So you will need to have notes in your the different tiers and kind of what that looks like. So the first one, the primary structure, is fairly straightforward. It's the sequence of amino acids. So here are some actual amino acids in this chain here, and we have a specific sequence of them. So the val is the valine, lysine, leucine, alanine, arginine, and they're put together, so there are 20 different ones we can choose from. You'll see that sometimes we have them repeated, sometimes they're close together, sometimes they're repeated far apart, and sometimes it doesn't have that amino acid at all. Okay, but the order that we've got will determine the shape. It will determine how it folds, and how it folds determines the shape and therefore the functions that it can perform. Then we've got our secondary structure. So the secondary structure is then how it folds together. So if we take our this is our molymod model of, this is a tripeptide, so I've got 3 amino acids together. So we've got our carboxyl group here, and so we've got our OH, we've got our double bond O, carbon-carbon, then we've got our nitrogen, and remember we lose a nitrogen and we lose the OH from this one to form the bond, the peptide bond between the nitrogen and the carbon, and then we've got the same here. So I've got 3 amino acids in this chain here, and these groups, so the carboxyl group here and the amine group here, they can interact with each other, and so this chain can twist around itself, so we can get this kind of twisting. Hopefully you can see that in the camera. And so when it twists around itself, we get what we call an alpha helix. So this alpha helix is the twisting of the chain, and it's held together by the interactions of the carboxyl groups with the amine groups. So if they're getting close together, they can then form a hydrogen bond, and the hydrogen bond will hold it in that position and stop it twisting any further. So we get that nice secure structure there. The other way it can fold, or it can shape itself, is by folding, and what we get is a beta pleated sheet. So if you imagine the zigzags here, so this is just one chain and it forms this zigzag, which you can kind of see in the picture here, so you can see the up and downs a bit like the model I've got here. Now if we get a strand of these and then it folds back on itself, let me show you with a wire. Oh, bear with me a minute. So if we've got our strand, if you imagine that this is a strand, so we can have it twisting to form our alpha helix like this, and these are going to be held together, so there's going to be hydrogen bonds between 2 sections of the chain. But we could also have the strands kind of folding along one another like this. Okay, so then we've got the strands running parallel or anti-parallel to one another, and we then get interactions between this strand and this strand. So we get hydrogen bonding happen between here, which holds them together in this kind of folded structure. And because of the zigzags, which obviously I don't have on my wire, but because of the zigzags, we then get what we call a beta pleated sheet. So some sections of a protein will be formed with this kind of sheet folding kind of thing going on, and other areas of the same protein might have some twisting going on. So we've got our alpha helix and our beta pleated sheet. So there's 2 different ways in which, as it comes out of the ribosome, it can begin to fold, it begins to take a shape. Okay, once it's done that, those smaller areas of folding can then fold round one another, and we get our tertiary structure. So our tertiary structure is then when we get interactions between the side chains of our amino acids. So remember, we've got that R group, and the R group can have different properties, and those different properties will affect how it interacts with the rest of the chain, but also the surroundings. So we're going to look at that in a little bit more detail and the types of bonding that we might have. And I've already mentioned the quaternary structure, and that's when we've just got chains bonding together. Okay, so let's have a look at some of these different bonds. So we've got hydrogen bonds, ionic bonds, covalent bonds, and hydrophobic and hydrophilic interactions. Okay, so the first type of interaction that we might get between our R group, so on my structure here, I just gave the R group, I just left it as an H. So we had two H's on this chain here and this chain here, which obviously just a hydrogen, that's fairly non-polar, and so we get kind of non-polar interaction between chains. So they might be like, oh, well, let's just come away from the water, guys. Okay, but this side group can have a sulfur atom in it. So if we have a look here with these two cytine molecules, so you can see we've got, oops, we've got a cytine here, cysteine here, and a cysteine here, and they've got a sulfur atom in both of them, and those sulfur atoms can both form a covalent bond with each other. And so if we've got two chains and we've got two cysteines, I'm just going to do it on my little tripeptide here, if we've got a sulfur here and a sulfur here, they can form a strong bond, a covalent bond, and that covalent bond is not going to break very easily, and so we're going to have this structure fixed in this place. So it's going to stay folded in that particular shape because of this strong covalent bond between these two molecules here, or two atoms here rather, sorry, these two sulfur atoms. So that's the first type of bonding, this disulfide bridge. Very, very important in some of our proteins. We've then got ionic bonds. So some of our side groups can actually form positive and negative charges. In the little picture here, you can see that we've got an amine group. Now this amine or amino group, NH2, we've met it here, if I show you on here, this is NH2, but it can gain an extra hydrogen, so it becomes NH3, but because it's got that extra hydrogen, it actually, which brings another proton in, it then becomes positively charged. So we get a positive charge on the amino group here, and the oxygen can lose the H, maybe that's where it went, and so it's negatively charged, and then we get an ionic bond between the negative charge of the oxygen and the positive charge of the hydrogen, and that holds them together. And again, an ionic bond is a strong bond. We've met those before, you've met those at GCSE, this strong bond between these two different side chains here. So this is a side chain, we've got CH2C, and then we've got the amine group here and a carboxyl group here, and they're interacting with each other. Okay, so again, that will be a strong bond. It's not going to break very easily. Water can begin to interfere with that because obviously it's polar. We've then got the third type of bond, are you ready? Is the hydrogen bonds, which we've already met. They hold them together in a secondary structure, but we can also see that here between this C double bond O and the NH here, we've got a hydrogen bond between the two. And the final type of bonding, just before I move on, just be aware hydrogen bonds can be broken. High temperature pH changes, particularly pH changes, is going to interact with this hydrogen bond, and it's going to cause that bond to potentially break, which can change the structure of the protein, which will then cause perhaps denaturing, particularly if it's in an active site. You've met denaturing, haven't you, at GCSE in biology lower down. The final type of bonding that we're going to meet is the interactions between different side groups that are hydrophobic or hydrophilic. Hydrophobic groups tend to stick together. So if you imagine that we have got in our protein, we've got our two hydrophobic groups. So here's my hydrogen side chain here, here's my hydrogen side chain here. They don't want to interact with water at all. So if we put this protein into water, what will happen is they will fold towards each other like this. So here I've got three kind of, hopefully you can visualize that, three folding towards one another away from the water. So in water, if you've got these hydrophobic side groups, it forces a bending and twisting of the molecule so that they are away from the water. And any of these more polar areas of the molecule will be outwards towards the water, and so we can end up with a kind of globular shape, this kind of ball-shaped folding in of itself. Because what you've got to remember is a lot of proteins are interacting with their environment, and the cytoplasm of a cell is mostly water-based. Okay, right, that is the overview of the key things that you need to know in this section. What I would like you to do now is to look at the PowerPoint, make notes on those different types of bonding. If necessary, read through the pages of the book. So the textbook pages you need for this are, and it's 3, I think, for this one. So you want to start on page 67, 68 and 69, which just kind of goes over the things I've just talked about. And then have a go at the 3 questions on the bottom of page 69 of this spread. Okay? If you've got any questions, you can email and ask. Otherwise, I will see you next time when we begin to look at some specific types of protein that we've got in our body. So we're going to look at the different kind of subgroups of protein, if you like. Okay, that's it from me. Happy note taking, and I will see you soon.